Hello everyone, welcome to the NPTEL online certification program on fundamentals of food process engineering. Okay. So, today we will start a new chapter on non-thermal processing, which is our last chapter of this course. By now we have discussed the various processing methods and preservation methods, and mostly they are by some thermal treatments. For example, we have discussed the sterilization, pasteurization, we have discussed the drying mechanism by which we can reduce the water activity and preserve the food for longer duration. There is evaporation, there are many techniques by which we can reduce the moisture content or water activity, and specifically we do sterilization and pasteurization to inactivate the microorganism by thermal treatments. But we have mentioned that because of this thermal effect, many important quality of the food may damage. For example, the flavor, color may hamper a bit, their quality may be hampered. So, there are new technologies coming into which are based on without heat treatment. Okay. So, we keep them in a category of non-thermal processing, and there are many non-thermal processing are coming into pictures nowadays. Some of them has been practiced in industry, and although there are high cost involvement is there in some of the non-thermal techniques. So, these are in constant improvement are going on, so that these methods can be used in a industry, food industry in a efficient way, and their cost of production may also decrease. So, in today's class and in the subsequent classes, we will discuss some of the non-thermal preservation techniques. So, basically the fundamental concept behind this non-thermal we will cover. In that, high pressure processing, which is one non-thermal preservation technique, pulse electric field technology, pulse light technology, irradiation, ozone and cold plasma technology, all this we will discuss. And at the end we will discuss a bit on Hudel technology, which is actually a combination of more than one methods, and combinedly those methods can cause very well inactivation of microorganism and can produce the safe and quality food product. So, let us start. So, first of all we can mention that food can be preserved by thermal or non-thermal process. Before the the invention of this non-thermal technique, the common perception was that for inactivation of microorganism only the thermal treatment will be effective. So, this thermal process such as heating and cooking affect the quality of the food in addition to inactivating the microorganism. So, they do inactivate the microorganism, but with that there are some heat sensitive element that also are getting affected. Conventional methods of thermal processing often produces a number of undesirable changes in the food. As I mentioned that color, flavor and functionality can be avoided. Okay. Even some nutritional component is there which will be damaged. For example, vitamin C, it is highly damaged because of temperature fluctuation or storage, etc. So, minimally processed food with better nutrient retention, high quality and fresh like food product, this is the demand of the consumer nowadays. So, they want a minimally processed food, so that the initial quality which we get during the harvesting will not be altered much, and the fresh like quality will be intact. So, the novel technologies that came in due course of time, when the scientists thought about some alternative way to preserve the food in a different way than the thermal processing, because there are certain advantage associated with that. So, first is retention of nutritional and sensory attributes, then the desired texture, sometime because of heat treatment the proper texture of the food will get damaged. Sometime shrinkage may happen that is not required, okay, or that gives some undesirable appearance to the product. They even form the some brown spot because of some browning action may have occurred there. And improved functional properties. So, this is also another desirable quality that we want to have. So, novel technologies have all these qualities. We can categorize them into these two segments. Okay. So, first is the electro technologies and the non-thermal technologies. In the electro technologies there are pulse electric field PEF, radio frequency heating, ohmic heating, etc. And in the non-thermal technologies we have high pressure processing HPP, membrane filtration, pulse light treatment, dense phase carbon dioxide and many more. Okay. So, first we will see high pressure processing. So, high pressure processing is not very new, we can say, because it has the first experimentation related to high pressure processing was invented around 1889, and that time it was not that much used, only it has been established that high pressure processing is efficient in decreasing the microbial contamination. Now, in around 1999 again scientists started on experimenting with HPP, so that to assess its effects on the food, basically apart from the inactivation of microorganism, what other changes the high pressure processing can cause, and still not much work has been done because of the high cost involvement of this high pressure processing. So, as the in the picture we can see that the total process chain of the high pressure processing occurs as first the unprocessed batch of the food. This comes into the chamber where we perform the high pressure processing. So, the product is loaded into the high pressure chamber. Okay. After that the vessel pre-filling is done. So, the water is entered from the low pressure channel to the chamber where it the pressure is intensified to a very high extent, okay, and the food is exposed to that high pressure. So, after this pre-filling the high pressure generation starts. Okay. So, this high pressure generation causes an isostatic pressure distribution. We can see from all the side the pressure is same pressure is applicable on the food product, and because of this mechanism the inactivation of microorganism and some enzymes occurs, and because of that the food will be intact since the pressure is from all the side we are providing. So, no damage, visual damage or the physical damage to the product does not happen, and it is very much in good quality, and then the high pressure is released, okay, and the product has been taken out. The process high pressure process batch has been taken out. So, mostly HPP is performed in a batch process or in semi continuous process also, but fully continuous high pressure processing has not been used and moreover it is not been used in food as well. So, this is how it works. Okay. Now, what is HPP and how it inactivates, how it works on the food products. So, it is a method, it is a novel method or non-thermal processing method which works on the principle of the Pascal's law, okay, and it also termed as hyperbaric pressure processing, as the pressure is much higher than the atmospheric pressure. also called the ultra high pressure processing, high hydrostatic pressure processing, Pascalization, etc. Okay. It retain the food quality and natural freshness and extend the shelf life. So, these all are speciality of this method HPP, and it is very much capital intensive process, because the initial setup that we require and this pressurization and depressurization, this cycle we have to perform for many times if we want to develop a system of industrial scale, and that has to run for years. So, we need to design a very robust system. Okay. So, capital intensive process it is, and although we can perform only a small batch in every operation. So, these are the limitations of the process. Potential application in processing of shelf stable products, blanching and pressure assisted freezing and thawing. So, there the application of high pressure processing is normally done. So, as we mentioned that it works on the principle of the Pascal's law. When an external pressure is applied at a point in a fluid contained in a vessel, it is transmitted undiminished and equally in all direction, right. So, this is actually performed that when we apply the pressure isostatically. So, the pressure this distributed uniformly in the whole chamber and around the food. So, from everywhere the equal pressure is exerted on the product. More than 200 years after Pascal's death, a chemist at the West Virginia Agricultural Experimental Station began studying the potential application of this law to the preservation of meat, juices and milk. So, we can see that this is the beauty of science, that when some invention when it occurs, we did not even think of that it can be used for food processing in an efficient way, but nowadays this is very promising technology, although it is cost intensive, but still since it gives a very good quality product. So, it has been mostly used in the juice processing and preservation, etc. So, HPP performs the sterilization of food without adding any additive to the food material. So, that way it is an additive free method, and it prevents the nutrient destruction, because no heat treatment we are applying to this. Also, because of tissue extraction, the extract allergens that will be destroyed and make the food product safe. So, the safety is the first concern that we can get by the HPP treatment. Second is the control of enzymes. So, by this method we can control the enzymatic activity, so that the food can preserve its original flavor and taste. For example, if we apply the thermal method, sometimes there are enzymes in the food itself. So, that can cause some decoloration, color degradation and off flavor development. So, those can be omitted in the HPP treatment. Okay. So, therefore, the nutrition is also intact. So, high pressure processing is applied to many cases in a food industry. For example, fruits and vegetable, then processed meat products, fish, then seafood, minimally processed food, then instant food, fruits and vegetable. So, all these are area including the sliced ham, salad dressing, rice cake, juices and yogurt. So, all these are the areas where the HPP is applied. Not in India, maybe it is not that much popular now, only for very high cost product preparation we use HPP. Although in a in the research oriented works are going on HPP, but in industrially not much utilization we have seen here, but in other countries it is in use nowadays. So, let us see how much high pressure is required to cause the degradation of the microorganisms or destruction of the microorganism. So, very high pressure is required here. The pressure used ranges from 100 to 900 megapascal, even 1000 megapascal in some cases, using 3 to 4 minutes to build up the pressure and holding time up to 5 minutes or more. Okay. And at a pressure of 4000 to 9000 atmosphere, enzymes and bacteria are inactivated, but taste and flavor remain unaffected. So, there are range of the pressure, not for every microorganism the destruction level or the pressure requirement for which they can be inactivated is not same. That may vary from microorganism to microorganism, even for bacteria or mold, those may vary, and generally the enzymes are resistant more. So, for them the high pressure is required, that is around 4000 to 9000 atmosphere. Okay. However, still the taste and flavor are not affected. So, we have seen we have a chart here that can show you that how the pressure varies from atmospheric pressure to autoclaving, hydraulic systems, then Mariana Trench, which is the deepest point where we can under the sea level in the world, where we can measure the pressure, and that is still the little higher than the 100 megapascal, and high pressure processing is even higher than that. So, you can imagine that how much strong system you have to build, how much strong pressure chamber you have to build for the processing of high pressure. Let us see the main component of high pressure processing. First, we require a pressure vessel and its closure. Okay. So, top and bottom closure and the pressure vessel we need. Then a pressure generation system, that is a very important component of the whole system, and the temperature control device. Although our target is to reduce the temperature, we do not want high temperature in case of HPP processing, but what happened that these are thermodynamic parameter, pressure and temperature, and these are have some synergistic effect. So, when we expose very high pressure, some increase in the temperature is inevitable and we want to control that. So, we provided, and sometime there are few application, not much in food, but in other application of HPP where temperature is also associated with the pressure. And we need a material handling system just to make the inflow and outflow of the product from the chamber. So, the material handling system is also required. Pressure generation in HPP can have by 3 different mechanism. Okay. So, for this much high pressure generation, either we can go for this direct compression method or indirect compression method, and the third one is the heating of the pressure medium. Now, let us see individually that what are these methods, the direct compression, indirect compression and the heating of the pressure medium. So, direct and indirect compression, if you can see the diagram A, which shows the direct compression. So, here the piston is used to direct pressurize the fluid or the pressure medium, which is here, okay. And there is a bottom closure will be there, the piston will be there, there is a press frame and a high pressure vessel. Okay. So, in the lab scale there is a this kind of system development is a bit tough because of the problem of sealing of the piston head and the chamber. So, there is a problem for generating this kind of a system in a small lab scale. Therefore, it is not much use for the food processing in a lab scale equipment. And there is another system which is the external pressurization or indirect type system. Here also the geometry is a bit same, that we have a press frame, we have a top closure and bottom closure. In the previous one we had only the bottom closure because from the top the piston is coming, and in this system we had the direct system, we have the intensifier within the chamber, right. However, in the indirect pressurization we have the pressure medium here, we have the high pressure vessel, top and bottom closure, and we have a different separate pressure medium tank, and we have an intensifier externally. So, this is fill the pressurized liquid is filled into this, and then the pressure is increasing indirectly by the intensifier. Okay. So, these two kind of systems we can observe, mostly this system is used in the laboratory scale or the batch food processing equipment. And the third one that I told, that heating of a pressurized medium. So, that is another method, because we know that when we heat the the pressurized medium, that the temperature will increase and also the pressure is increased, that pressure is increased in terms of a vapor pressure actually. So, that is in this particular case we are not going to use that for HPP, but that is also one method. Now, isostatic pressing system. Isostatic pressing system can also be of three type. First is the cold isostatic pressing, also this is called the CIP system. So, cold isostatic pressing system is this diagram, where the product is there, okay, and then the pressure is from all the side, because it is isostatically felt by the product that we are keeping inside the pressurized medium. So, whatever pressure we are providing, that pressure from the pressurization source, that is uniformly distributed on the flexible mold. Okay. Now, in the other case, when we see the warm isostatic pressing system, there we have a heating element, okay, we have a heating element, and across a two electrode we are sending the electrical current, and that heat this section. There is a thermocouple attached which control the temperature of this two heating section, okay, and the food is or the material is inside the pressurizing chamber. So, this is warm isostatic pressure. Okay. So, this is another method, and the third method is the hot isostatic pressing system, where we have a heater inside adjacent to the media, and there is a heat insulator, and the inert gas atmosphere is there, okay, where the heater is. So, in this kind of a system we provide the hot isostatic pressing system. So, that means with pressure we are having the effect of the high temperature as well, but normally for food we are using the cold isostatic pressing systems. Modes of high pressure processing, as I mentioned, that we mostly get the batch processing module, and somewhere we can see the semi continuous. We hardly get the continuous module, specifically for food we get very less. So, mostly these two we can observe, and this is the batch mode kind of a system, where there is a intensifier externally and there is a water inlet So, this high pressure pump is sending this pressurized water into the chamber. After the loading of the product is done, there is a pressure closure and there is a heating or cooling functional option is given, okay, and the water outlet is there. When we want the release of the pressure, that time there should be some exit of this pressurized water. So, there is a pressure release valve and the water outlet. So, this is kind of a system which is called the batch HPP system. So, what is the other system? The other system is the semi continuous mode, when what happens is that the liquid food we are sending by pump to the chambers and there are chambers in series. Okay. So, this is entered into this pressurized the liquid food is entered into the chamber, okay, and then after one state of processing, this is entered to the process liquid section, and the since it is in series. So, for all the consecutive pressure chambers, the feed is entered. Okay. So, this is entered here, okay, and then it is going to this channel and then entered. So, like that it is going, and the after processing, this is coming to the common exit channel. So, the process liquid is going out, and the high pressure pump is there which is sending the pressurized liquid here, okay, and that is again sending to the other consecutive lines. Okay. So, what happen is we make a sequence here. When one chamber is in pressurized condition, the other chamber is in the depressurized condition. Okay. So, this sequence is maintained in all the condition, all the consecutive section, and that we call a semi continuous system. So, we continuously feeding the liquid food in all the chambers, and we maintain the sequence of making one as pressurized, the other as depressurized, and this sequence is going on and the semi continuous operation is performed. So, to summarize the process, first the packaged food in a sterilized container is given, then that is loaded to the HPP chamber, then fill the pressure chamber with water, pressurize the chamber, this is done by the intensifier, then hold under that pressure for a required period of time, then depressurize the chamber and remove the processed food. So, this is the whole chain that we perform in HPP system. Now, the most important thing, that is what is the effect of high pressure processing on microorganism. Okay. So, microorganism which in the atmospheric pressure can very well sustain, as we increase the pressure up to 250 megapascal, they start I mean their growth stops and they start to decreasing. So, then what happened, that when we increase the pressure up to 600 megapascal, so the complete inactivation or the destruction of the microorganism will be there. So, at 250 they start inactivating, and at 600 megapascal they fully destroyed. So, when we release then the pressure and bring it to the normal condition, the microbes will not be again can grow and contaminate the food. So, the lethal effect of HPP on microorganism is due to the several changes occurring in the microbial cell. So, if we see the microbial growth rate versus pressure, so the growth rate per hour versus pressure, if we can see, so as we increase the pressure, this is in megapascal. So, as we increase, the normal bacteria will destroy as we reach to the little bit higher than the 40-45 around. Okay. So, some bacteria are there, they can withstand little bit of pressure. So, those are called the piezo tolerant. Okay. There are piezophile, so which can sustain even up to higher, that is around 100 megapascal. Okay. And obligatory piezophile, these are, there is a range of this bacteria. So, mostly they will destroy beyond this pressure level. So, if we see the destruction kinetics of the microbe, as we have seen, we have discussed this in some of our chapter in this course, that microbial death kinetics, how from the initial number of N0 to N they are coming with a time t and how they decrease. So, here also the same principle is valid, only the difference is that in that case the destruction was because of the thermal effect and here the destruction is because of the pressure effect. Okay. So, if we plot that the initial number of microorganisms with time, we can get the equation as this, ln n equal to ln n0 minus kt. So, log n by n0, that is equal to log of st, that is equal to minus t by d. So, this is called the sterilizing value, if you remember. So, this way we can calculate where d is the decimal reduction time and t is the time taken to inactivate the microorganism. And z, we know that z is the is that by in case of the thermal effect, we have discussed that z value causes the increase in the temperature, for that one log cycle reduction will be there. Okay. So, this is the effect, that z equal to 2.303 R T square by E A, and if we know the two temperatures, so z is equal to T2 minus T1 by log of D1 minus log of D2, where D1 D2 are the inactivation d value of temperature corresponding T1 and T2. So, in this case, the destruction follows this kind of a pattern, which will be explained by the Weibull distribution model. So, this is one model, there are many such kind of model exist in high pressure processing, that has been generated by empirical model development. So, St is equal to exponential minus t by alpha to the power beta, where Weibull reliability plot, if we draw, where beta varies from 0 to 1, which is this plot, beta equal to 1, okay, and beta greater than 1. So, for that it has been plotted as Weibull reliability with respect to time. Okay. So, this equation S t equal to exponential minus t by alpha to the power beta can be simplified as log S t, that is equal to minus 1 by 2.303 into t by alpha to the power beta. Reliable life, where the 90 percentile of failure time distribution can be expressed as T r, that is equal to alpha into minus ln 0.1 to the power 1 by beta, that is equal to alpha into 2.303 to the power 1 by beta, and the d is the number of decimal reduction. So, number of decimal reduction T d, that is equal to alpha into minus ln 10 to the power minus d to the power 1 by beta. So, this is how the destruction, the microbial destruction can happen, okay, for the different beta value, and these values as depends on the average shape parameter, that is the beta, and the alpha is the calculated scale parameter. So, how that can be interpreted, that Weibull distribution model. So, as we mentioned that there is a pressurization initially, then we have to hold it for a certain time and then again release of the pressure will be there. So, here in this pressure and temperature diagram, this diagram we can see 1 to 2 the pressurization effect or the pressurization will occur. So, when we have reached time scale here, then from 2 to 3 there is pressure hold. So, we will maintain the pressure whatever we have achieved at P2 up to this time, okay, T3, and then again it reduces to the atmospheric pressure. So, this two effect we can categorize into two different graph. One is considering the pulse effect, that is increase of the pressure and decrease of the pressure. Okay. So, this is that, and the other is showing the isobaric kinetics, that is when constant pressure, isostatically constant pressure was applied on the food, that time what was the degradation. So, this is initially we had Ni okay, initially we had N0 microorganism, okay, sorry, here Ni is the initial and No that we are attaining after certain time. Okay. So, when we reach to No, then No to Nt, initially it was Ni, then we have reached to No and No to Nt that we hold it, okay, and after that the degradation will be. So, to model this part, that is the pulse effect, the equation is pulse effect equal to minus log 10 N0 by Ni, and the other case the isobaric kinetics, that effect is visible as log 10 N0 by Ni equal to minus B T to the power n, right. Okay. So, altogether when the destruction is from the initial to the final one, so minus log 10 Nt by Ni, that is equal to this effect, pulse effect N0 by Ni log 10 minus the other isobaric kinetics. So, this together, so we can write Pe plus Bc into T to the power n average, where n average is the average shape parameter and C is the calculated scale parameter. That means, the alpha that we have discussed there, and n average is actually the resembles with the beta factor. We can write this, the calculated scale parameter Bc. So, log 10 Bc, that is equal to T by Zt plus C2, where Zt shows the temperature resistivity and log Bc to the base 10 equal to P by Zp plus C1. So, here Zp signifies the pressure resistivity. Okay. So, temperature resistivity and pressure resistivity, these two are the parameter by which we can assess that how much the destruction of the microorganism will takes place, and using this combined equation we can assess that what will be the destruction of the microorganism. So, this is how the Weibull model is interpreted. I hope we have reached almost the end of this HPP. So, let us just summarize the advantage of HPP. It is not dependent on the size and shape of the food, and because the shape does not matter, in the sense if you apply the pressure, it is applicable on both the sides. So, all the particle of the food or all the section is exposed to the same pressure condition. High pressure is independent of time or mass, that is it acts instantaneously, thus reducing the processing time. So, it is not that suppose temperature you are giving and gradually the temperature is increasing from the, or temperature has to reach from the surface to the center, but here the pressure is instantly distributed throughout the whole section. It does not break the covalent bond, therefore, maintain the natural flavor of the product. It can be applied at room temperature, thus reducing the amount of thermal energy needed for food products during conventional processing. Since high pressure processing is isostatic, the food is preserved evenly throughout without any particle escaping the treatment, and the process is environment friendly since it requires only electric energy and there are no waste product generated. So, that way it is very helpful. It has some specific effect on the proteins and enzyme, inhibition or simulation of enzymatic activity. So, it depends on the processing condition, that how much pressure we are providing, how long we are providing. Proteins are partially denatured in products where protein have not been previously modified by other process such as heating, drying and fermentation, etc. So, protein are partially denatured in these cases. Pressure affects hydrophobic bonds and electrostatic interactions. Tertiary and quaternary structure become affected, whereas the secondary structure become affected over 700 megapascal pressure. So, we can see that when unfolded structure enters into a HPP chamber and we provide the isostatic pressure, it may happen that the condition A arise, that is release of the pressure will cause denatured structure of the protein, or it may happen that the the release may cause the unfolding from the unfolding state to the native state conversion will be there. So, native to unfold can happen because of the application of pressure, and again the unfolding to native may also become because of the release of the pressure. Okay. But in some cases when denaturation happen, so there it is not an reversible process. Okay. So, native to unfolding, this part is reversible, however, from the unfolded to denatured part, this part is not reversible. Okay. So, the limitations is that food enzymes and bacterial spore are very resistant to pressure and require very high pressure for their inactivation. Okay. So, spores, inactivation of spore and enzyme require very high pressure. The residual enzyme activity and dissolved oxygen results in enzymatic and oxidative degradation of certain food component, and most of the pressure processed food need low temperature storage and distribution to retain their sensory and nutritional quality. So, we will stop here and we will continue with the next topic in a next class. Thank you.